Well, I should thank you for being here, and I will tell you about a morphogene, which is called OTX2, and the work has been done in the lab by, the work is done in, no, this is not the slides yet, I'm sorry, I was worried about that, okay. So, we're working in the lab on signaling transcription factors. In fact, we discovered something like 20 years ago almost, I mean, that homeoproteins are not only cell-autonomous transcription factors, but are also non-cell autonomous transcription factor, also translation regulator and epigenetic modifiers. And the transfer of the cell, or the transcription factor between cells depends on two sequences which are in the homeodomain, in the DNA binding domain, are very, very highly conserved between all these transcription factors. Although we have not tried all of them, I mean, all those that we have tried can do it in vitro or in vivo. So this has been done in many systems by us and by others. Plants do it, and this is well known, and flies as well. This was a collaboration between our lab and Florence Macha in Montpellier. Fishes do it, and this is also collaborative work. Frogs and chicks do it, and this was in collaboration with Christine Hart and Bill Harris in Cambridge in UK, where we work on axon guidance and the role of the transfer of engreled in the anterior-posterior patterning of the tectum. And mice do it, and this was a collaboration that we did with Takao Hench, and there are also some independent studies on the same theme. So these transcription factors, as you know, are expressed during development, and this is something that everybody knows, but they are also expressed in the adult. And as you can see, the patterns of expression during development is not the same, it's very dissimilar. And in fact, I mean, if you look at OTX2, which will be the protein we're going to discuss today, I mean, very early in development, it is in the anterior neuroectoderm in the brain, but as the animal develops and ages, I mean, the expression in the cortex is totally lost, and you have a new pattern of expression in other regions. Now, the functions of these transcription factors, I mean, in the adult, and the function of signaling transcription factors in the adult is more or less unknown at this point. Now, one of the big issues that we had is in discussing this separation between cell-autonomous and non-cell-autonomous functions in these transcription factors is that the sequences which are responsible for getting in or getting out are in the homeodomain. And this means that you cannot mutate them. If you mutate them, I mean, you lose the cell-autonomous activities, or you modify the cell-autonomous activities. And this led us to a new strategy, which was to design nanobodies based on the linking of the light and heavy chain variable chains with the hinge and the signal peptide. And in fact, Alessandra showed you the use of these single-chain antibodies. And to make mice, to fabricate mice that can induce, in which you can induce the secretion of these antibodies and block the protein outside so that you only modify the non-cell autonomous activity of the transcription factor. And I will give you some examples of this strategy, I mean, to separate the cell autonomous versus the non-cell autonomous activity of homeoprotein transcription factors, in this case, OTX2. But we have mice also for PAC6, as you've seen before, and for engreled. Now comes the slide. I'm going to tell you of a work that we've done in collaboration with the Takao Hench lab. I mean, at the time, he was at the Riken Institute when we started. Now he moved to Harvard Medical School. And Sayaka Sugiyama did the first experiments in collaboration with people in my lab. These are the people in the lab who've been still in the lab, in fact, and some have left. I mean, all these people have left now, and I'm going to show you the work done by Jessica, Namsook, and Ariel, and Jérôme, and Damien as well. And indeed, Michel, who's been working with me for almost ever now. And it will, I hope it will continue like that. Not forever, I hope. Okay. Now, cerebral cortex plasticity is a matter of the ability of the brain to learn, okay? I mean, contrary to what you think, and many of you think, I mean, there's not a you don't learn throughout life. There's a short period of time during which the brain is able to learn, and this short period of time during which the brain is able to learn is called the critical period, for obvious reasons. But the critical periods for the different types of learning are not synchronous. For example, learning something like social skills will start pretty late in life and will continue for almost ever. Language start early and will go down, and the ability will go down. The sensory systems, you learn very early, so the visual system, the hearing system will start early and will close pretty early. So in fact, there's a period during which you cannot learn, a period during which you can learn, and a period during which you can not learn anymore or very little. Now this is called neuroplasticity. This neuroplasticity, you can see it here. This is for example, the visual system. So in a mouse, for example, if you close one eye at, this is binocular vision, so you can close one eye, and during the critical period, during the plasticity period, if you close this eye, the terminals coming from the other eye through a thalamic relay will invade, the synapses, which are left free by the non-use of a closed eye. So it's a plasticity, a morphological plasticity that you can follow that way. And this is what you see here. For example, if you have no monocular deprivation in a mouse, can I see? Yeah. In the mouse, you have an advantage of the contralateral eye because the eyes are slightly on the side. If you close the eye at P18, you have barely no shift in the equilibrium between the contralateral and the ipsilateral eye. But if you close the eye at P25 during the critical period, then you can move the dominance of the system so that you shift toward the ipsilateral eye. And this is due to the maturation of these little guys here called parvalbumin neurons, like synapses on the pyramidal cells. And most of the plasticity period accompanies the slow maturation of these parvalbumin cells. This is shown here. This is these large basket cells, that synapse on the pyramidal cells. And as they mature, as you can see here, this is before critical period. They are present, okay, and they are generated from the ganglionic eminences at E15 in the mouse. So they are present in the cortex, but as the cortex matures at P30, now they are fully mature, and this is the period of plasticity. And when they are fully inhibitory, fully mature, you close plasticity again. And as you can see here, this is the parvalbumin neurons at P19. So before the opening of plasticity, they are not very well differentiated. Now, at the peak of plasticity, P29, you can see that they express parvalbumin a lot. What you can see also is that they express OTX2. Well, they express OTX2 is not the word I should use, but they have OTX2 you have OTX2 in the PV cells, and not before the critical period, but at the time of the activation of plasticity. Now, in fact, the surprising thing is that this OTX2 is not made by the PV cells. If you look by inside hybridization, the OTX2 GFP mouse, which was given to us by Antonio Simeone, you don't see the locus is not active. You see the protein. It really is a protein. You can verify by mass spec. And if you recombine the locus using a driver which is not expressed in the PV cells, you lose OTX2 in the PV cells, and also you lose the maturation of the PV cells. So in fact, you have the protein, but the protein is not made by the cells. So when we saw that with Takao, we decided to we saw this, we said, well, maybe the maturation of the PV cells, the opening of plasticity, is due to the arrival of OTX2 into the PV cells, which was indeed surprising, but we decided to test the hypothesis, and we infused OTX2 in the cortex. So if you take OTX2, now you infuse OTX2 in the cortex, it diffuses throughout the cortex. And as you can see here, this is non-infused side. This is before the opening of the critical period, it's P18. So you can see that at P18, you have no OTX2 in the PV cells, or very little. The PV cells are not differentiated. If you infuse OTX2, it is internalized by the PV cells, and now you provoke the maturation of the PV cells. At the same time, if you do that very early, you can see that you open plasticity early. This is a shift which is due to plasticity at P18 now with OTX2, which never happens in the vehicle, as you can see here. But at P25, you close plasticity earlier. And in fact, if you remove OTX2, you do not open plasticity at all. So in fact, what we see here is that the arrival of OTX2 is necessary to open plasticity at P20 and necessary and sufficient to open it at P20 and close at P40. If you remove OTX2, you don't open plasticity before or later during development, as you will see. And if you infuse OTX2, I mean, you can open plasticity and close it earlier in time. So in a way, to go back to the theme of the meeting, it should remind you of the French flag problem. In fact, in the Lewis-Wolpert's model, you have the gradient, and you have thresholds of expression of transcription factors due to the, well, this is a linear gradient in this case, which is really the case. And here, I mean, you could say that you also have a French flag, but it is at the unicellular level, and is due to the concentration of OTX2 within the cells. Below a certain concentration of OTX2, you don't open plasticity, and you reach a second threshold of OTX2 within the PV cells, and now you close plasticity. And to start the accumulation of OTX2 in the PV cells, you need a sensory input, as you will see. So a very surprising thing, I mean, we were very surprised by that with Takao when I went to the experiment was done in Tokyo at the time. So I went to Tokyo and discussed with him, and we are extremely surprised to see that we had a specific internalization of OTX2 by the PV cells, because this meant that we have binding sites expressed by the PV cells, the binding sites for a transcription factor. So we did some staining in the cortex, and we could see that, in fact, OTX2 is primarily within cells, the PV cells, which are surrounded by complex sugars. I mean, in fact, by what we call the perineuronal nets, discovered by Camillo Golgi almost a bit over 100 years ago. And as you can see here, I mean, there's a correlation between the presence of this matrix, which can be decorated by a specific lectin, and the presence of OTX2 within the cells, suggesting that possibly, I mean, the sugars were providing the binding sites for the transcription factor. So indeed, this led to the next experiment, which was to remove the sugars. So you can remove the sugars by injecting a concatenase ABC. You see, you remove the sugars, you don't have the lectin binding anymore, and you can see that you have a reduction of the endogenous protein in your PV cells, suggesting that if you remove the sugars, the protein doesn't find the PV cells as well as it should. So we identified the domain which in OTX2 is responsible for the recognition of the sugars. The sugars are scoring sulfate E and D with 2 sulfates. We don't know yet whether it's E and D. We think it's E, and we've not totally, I mean, demonstrated that. But what it shows that this RK peptide, which is a little domain here, also in the homeodomain, almost in the first helix of the homeodomain, is responsible for the binding to the sugars and to the binding to the PV cells at the same time. So that if you infuse the RK peptide in vivo, you compete OTX2 away because you block the binding site of OTX2 on the PV cells, and this does not happen with a scrambled peptide or a mutated peptide with 2 alanines instead of the RK doublet. Because the scrambled peptide doesn't do it, it's a sequence-specific recognition of the sugars. And when you do that and remove OTX2 by competition with the RK peptide infusion, you see you have less WFA binding and less parvalbumin expression, as if you were rejuvenating your PV cells. And in fact, you can see that you reduce OTX2 content, you reduce WFA staining, you reduce PV expression, you rejuvenate your cells. This is in an adult mouse. It's an adult mouse. And because you rejuvenate your cells, you may say, well, do I reopen plasticity if my neurons are young now, okay? And indeed, this is the case. You can take a mouse, and you can make the mouse blind from one eye. It's amblyopia, in fact, as you can see here, by closing the eye during the critical period. And if in the adult now you infuse a peptide, you reduce PV expression, you reduce the PNN assembly, And as you can see, if you look at the visual acuity, now you restore visual acuity in your mice. So this is probably one of the first demonstrations that you can cure a neurodevelopmental disease by blocking the internalization of OTX2 by the PV cells. Now, the big question is, if we have OTX2 in the PV cells, and if the PV cells don't make OTX2 themselves, where does it come from? Okay, and it comes from several possible places, but one place is very likely the most active one, is the choroid plexus. Now, the choroid plexus is a little organ that secretes the CSF in the brain, and you can see it here, here, and here. I mean, there are several regions where you have choroid plexus, and the choroid plexus expresses OTX2 throughout life. You see it by in situ hybridization here, you see it by immunohistochemistry, you have OTX2 in this monolayer of cells, and you can recombine specifically OTX2 in the choroid plexus by a trick that I'm not going to discuss. If you do that, you remove OTX2 from the choroid plexus at the messenger RNA and the protein level, and what happens? Well, what happens is that we are still in the adult. What happens is that indeed you reduce OTX2 levels in your PV cells. You demature, so to say, the PV cells. You lose you decrease PNN assembly. Nothing happens to the calreticulin cells. And indeed, you have now, after a few days, 2 weeks, in fact, because the protein is pretty stable, you have the level in the PV cells of OTX2 that you have during plasticity, during the critical period. So the protein comes from the choroid plexus, travels through the parenchyma, finds the cells, gets inside the cells, and initiates the maturation. And in the adult, maintains the non-plastic stage. Since you remove it in the adult, you reinstall plasticity. It means that you need the permanent internalization of OTX2 to be non-plastic. Now, if you do that in the choroid plexus, as you can see here, you can look at the visual acuity again. In this case, what you do, you remove OTX2, you have a normal mouse, okay? I mean, a healthy mouse. This is not a very nice experiment because the mouse is very happy. It sees with the 2 eyes. And now you block OTX2 by recombining OTX2 in the choroid plexus. So you reduce OTX2 in the PV cells, and you blind the mouse, and it becomes amblyopic. So you instill the disease in the adult, which normally does not happen because in the adult, normally, the system is not plastic. Well, you can do the other experiments, which is nicer. You have a blind mouse, blind from one eye, a lazy eye mouse, and now you reopen plasticity in the adult, and you restore visual acuity. So just as this leads to the two, I'm sorry. This is to the two-threshold model for cerebral cortex plasticity, where you have the sensory input. The sensory input, you start the OTX2 accumulation. First threshold, I open plasticity. Second threshold, I close plasticity. But in the adult, if I remove OTX2 acutely, I reopen plasticity by getting into this region here, where I am plastic. I say you need the sensory input, and in fact, you need a sensory input because if you did not need a sensory input, all plasticity periods will be synchronous. Okay, there's no reason why you should open plasticity in the eye at a certain period of time and open plasticity in the auditory cortex at another period of time, or other cortices as well. And if you dark clear an animal, for example, so you close the eye of the animal will never open plasticity, and but if you infuse the TX2, you will force the maturation of the system, even though the animals are dark cleared. So how is it possible that you need the sensory input to open plasticity? And this means that the activity of the eye allows plasticity opening, allows the internalization of the TX2 by the PV cells. So this is still a bit under study, but in fact, you have also a TX2 in the eye. The TX2 is expressed in the bipolar cells and transferred from the bipolar cells to the ganglionic cells that project onto the thalamus. Now, if you remove a TX2 by injecting siRNA linked to a cell-penetrating peptide, then what happens in the eye is that you block the opening of plasticity. The siRNA in the cortex does nothing because there is no synthesis of a TX2 in the cortex. And the antibody, if you inject it in the eye or in the cortex, will block the opening of plasticity. So that, in fact, Carol Mason and Alexandra Repsan proposed in a News and Views that what happens is that you have the bipolar cells that send a TX2 into the retinal ganglion cells that will now travel to the dorsal thalamus. You also may have some TX2 coming from interneuron in the dorsal thalamus that will gain access to the PV cells, allowing the first assembly of the PNNs. And now that you have the PNNs assembled, OTX2 surrounding the PNNs will get inside very easily because of the first assembly, which is activity dependent. In fact, we don't know whether this happens, but if you inject a TX2 in the eye, you find it into the PV cells after several synaptic relays. So it can travel along the axonal pathway, the visual pathway, to gain access to the PV cells. Now, when I used to discuss these things, I mean, it was not always, well, I mean, it was very intriguing, let's say it that way. And in fact, the geneticists say, well, you need the genetic evidence. I mean, as long as you don't have the genetic evidence, well, it's not true. You know the geneticists. So we decided to go for the genetic evidence, okay? The first genetic evidence that we could have was coming from this this RK peptide. So we generated a mouse in which we replaced the RK peptide by a doublet of alanine. And as you could see, I mean, this protein now with the AA instead of RK doublet, just upstream of the first helix of the domain. Now, this protein in the animal is internalized by the PV cell still, but it becomes promiscuous because it does not recognize specifically the PV cells. So it goes into many cells, and the accumulation of the protein is very much diluted, okay? So, I mean, it does not increase as fast as in the wild type within the PV cells because of the fact that it goes into other cell types. Now, the consequence of this dilution of the TX2 between cells is that the maturation of the PV cells is slowed down. I mean, instead of being mature at, say, P30, P40, like you can see by the assembly of the PNNs with the WFA staining, it takes 100 days before they get the maturation. And this is the same thing for the PV maturation. And in fact, if you look at plasticity, in this mouse now, you don't have plasticity at P30, but you have plasticity at P100. So you have to wait until you have enough OTX2 within the cells to open plasticity. The other genetic option is to use the single-chain antibody mouse, where you can, now you have this single-chain which is inducible. So it's a Cre-inducible antibody. You can cross the SCSV OTX2 mouse with the PV-Cre animal so that, in fact, the cells that make parvalbumin will secrete the antibody and will, as soon as parvalbumin starts to be expressed, will secrete the antibody and block OTX2 outside. And if you do that, I mean, what happens is that you decrease, you retard the arrival of OTX2, you retard the maturation of parvalbumin and the assembly of the PNNs, and the plasticity genes, ARC, FOS, NUR1, and AGR4, are not activated as they should be in a normal mouse, or in a mouse in which you have activated the PAX6 antibody, which has no effect, indeed, on OTX2 arrival. Now, you can also do that in the adult, and in the adult, you can force the secretion of the antibody in the choroid plexus. Now, if you do that, what happens in the adult? You have the expression of the SCSV OTX2 antibody. Now you up this one, you up-regulate the activity, the plasticity genes, as you can see here, and you reopen plasticity in the adult. So this is the genetic evidence that, in fact, by blocking OTX2 transfer inside the PV cells, you will regulate plasticity, either block it at young ages or reopen it in the adult animals. Now, this is true for the visual system. It's not only true for the visual system. It's true for the entire cortex. You see it in the auditory cortex. And this was validated also for the medial prefrontal cortex. I won't show you the data. It's a work done by Ariel DiNardo in my lab. And in fact, I mean, it's also true probably for most higher cognition systems. And this is very interesting because, as you know, I mean, in psychiatric diseases, the critical period comes very late, by the age of 15, where you have the increase in inhibition, which is due to the maturation of the PV cells in the anterior cortex, and to the decrease in the excitatory synapses also in the prefrontal cortex. So we hope that this system is also acting for functions which are as interesting as visual functions or auditory functions. Now, I will finish with the epigenetic evidence, which I think is also something which is rather interesting. In fact, we wanted to identify which are the OTX2 target genes, I would say the non-cell autonomous transcriptional targets of OTX2 within PV cells. This was made not easy, but simpler by the fact that you have, at P30, you have plasticity in the wild-type animals. In the OTX2 heterozygote, at P30, you have no plasticity. But in the wild-type, in the OTX2 GFP, you have now plasticity at P100, but in the wild-type, you don't have the plasticity at P100. So by doing RNA sequencing experiment between the wild-type and OTX2 GFP mouse, the heterozygote mouse, at 2 ages, P30 and P100, it was sort of easy to find genes which are only regulated during plasticity in the PV cells, okay? And we ended with a list of something like 21, 22 genes, you can see here. And indeed, you find, I mean, the suspects that you would expect, like ARC, EGR2, FOS, NUR1, etc., okay? These are the genes which are upregulated during plasticity. Does not mean that they are direct targets of OTX2. So to see whether they are direct target of OTX2, we infused cyclohexamide at the same time. Now, if you include cyclohexamide, you see there are only 2 genes which are really upregulated. And these are GAT45V and GAT45G. And you can see here, in fact, during the critical period, they are upregulated and downregulated after. So it is as if, in fact, you only have one locus, which is a target locus of OTX2, during the critical period in the PV cells, and associated with plasticity. There might be other ones, but associated with plasticity, and this would be these GAD enzymes, which, as you know, are demethylating enzymes. Now, we verified that indeed GAT45 is a direct target. We found 4 sites which are recognized by OTX2. We verified it by the jet shift experiments and chromatin immunoprecipitation experiments. So we believe that GAT45 is really a direct target of OTX2. And indeed, I mean, if this is true, it means that in all situations where you have plasticity, you should have GAT45 overexpression. So what we did was to take the adult mouse and to block OTX2 by expressing the single-cell antibody against OTX2. And when you do that, as you can see, you know that you reopen plasticity. I just showed you that before. And if you do that, you can see that now you have an expression of GAT45. Not with the PAC6 mouse, but only with the OTX2 mouse. So, I mean, so the morphogens, so to say, will now upregulate GAT45 activity, I mean, reducing OTX2. You can see that GAT45 is expressed in layer 4, and we have quantified it, layer 4 and layer 5 and 6. So you have a specific overexpression of GAT45 in a situation where you reopen plasticity in the adult. If so, infecting the PV cells with GAT45 in the adult should reopen plasticity. And indeed, this is what happens. As you can see here, you can express GAT45 using a viral vector in the PV cells. So you do that, you have GAT45 in the PV cells. And now you can see that you have a correlation between the expression of GAT45 and that of plasticity genes, ARC, FOS. I'm sorry, this is doublet, doesn't matter. But you can see here that you have this overexpression of the plasticity genes and that you have also reopening the reopening of plasticity. This is associated with modifications of the methylation on the promoters of the plasticity genes, not ARC, because ARC is not in PV cells. In fact, ARC is in pyramidal cells. But in the these plasticity genes, you can see that you have a modification of the methylation of key CPGs in the promoters. Whether they are the ones that count, we don't know. We need to modify them using CRISPR-Cas9. This is not done yet. It will be done. Now, the fact that GAD45 regulates the methylation of in the genomes led us to look at the modification of methylation by looking at CpG staining in the nuclei. And as you can see here, I mean, between P20 and P100, as the cells mature progressively, because of the internalization of OTX2, you have a modification in the pattern distribution of MESCP2 foci. I mean, at the beginning, you have 4 foci, and I mean, stepwise, you decrease the number of foci, and this is specifically inside the PV cells. It's not in non-PV cells. I mean, outside of the cells decorated by the lectin, you see nothing happening. And this means that the increasing concentration of OTX2 within your nuclei slowly modifies, probably stepwise, the structure of the chromatin, probably giving access to different target genes within the chromatin. This is, you see this. I mean, you also have it when indeed you re-express plasticity in the adult by forcing the expression of the OTX2 antibody. And you see it also when you overexpress GAD45 when you reopen plasticity. So in fact, the number of MESCP2 foci is really correlated with the activity status, the plasticity status of your cerebral cortex. And the same is true for other marks, epigenetic marks, for example, H3K9 methyl-3, and also for DNA strand breaks, as you can see by the staining with gamma-H2AX. And this might have something to do with the expression of long genes, but we're still working on that. But each time you reopen plasticity, you have a change in the epigenetic marks, which is parallel to what you see during development. So I'll show you this summary slide in a way. So you have this sensory input. I mean, the sensory input forces the first assembly of the PNNs, allowing the first accumulation of OTX2, first threshold, I open plasticity, second threshold, I close plasticity. If I reduce OTX2 by several means, for example, RK peptide or the expression of the nanobodies, or if I express GAD45 in the PV cells, I can reopen plasticity in the adult. And this is paralleled by the expression of GAD45. Well, here it's obvious, but in this case, before plasticity, when you open plasticity, you upregulate GAD45. When you close plasticity, you downregulate GAD45. You modify your decoration by MESCP2 antibodies. And if you reopen plasticity in the adult, indeed, you go back to the plasticity system. So that's where we are. I would just like to thank the people that have given the money to do the work. I mean, as you can see here, I mean, the classical, I mean, the usual suspects. And I will thank again my colleagues. In fact, Jessica and Suk and Ariel have worked a lot on the targets and the later aspect of the work. Clémence, Marine, Julien, and Clémentine did start the work, and Clémentine did a lot on the prefrontal cortex, the medial prefrontal cortex. She's more in the behavior, I mean the psychological behavior of the mice. I had no time to present her data, but she's working on that. All these guys have left. I already talked about Michel. This is Takao. And Sayaka again, who initiated the work with Takao and me, in fact, and that's more or less it. I would also like to thank all of you, because you came from very far sometimes, and we had 2 very good days, and at this point, I'm ready to take questions.